Technically, we're 96% identical to our evolutionary cousins, which is quite amazing when you think about it. What's also amazing is that we share 70% of our DNA with a slug and 50% of our DNA with a banana.
This is both humbling and confusing. I mean, how could we humans be so complex if we have so few genes? And they're mostly the same as those of our closest living non-human relatives. Well, a few years later, in 2005, the Chimpanzee Genome Project also produced its first draft sequence. Having both human and chimp genomes, as well as other more distant relatives, like mice, allowed us to compare not just our genes, but our non-coding regions too. See, some experts suspected that there were sequences hidden in the junk DNA that were actually functional. Not because they directly coded for proteins like genes did, but because they somehow regulated the activity of those genes. And changes in those regulatory sequences that were unique to humans could be just as important, or even more so, than changes in the genes themselves. And in 2006, researchers identified around 5% of the human genome that was non-coding, but tightly conserved across species, which means some of it probably plays important regulatory roles. Think of these sequences as switches that control the activity of genes. And those switches have been around in the non-coding DNA of vertebrates for a long time, and are generally very similar between species. But in humans, thousands of them have been tweaked, altering the way they work and how they control our genes. Scientists call them human accelerated regions, or HARs. Many of them are associated with brain and nervous system genes, changing how those features develop compared to our closest non-human relatives, like chimps. Okay, so search over, right? Humanity comes down to some mutations in the 1% to 2% of the genome that's made up of genes, plus a bunch of tweaks in the 5% that regulates genes. Job well done, everyone. We can go home. Well, not quite. In a paper published in 2022, researchers announced that they had found yet another previously unknown layer of the genomic onion. They expanded their search to the rest of the genome, the more than 90% that is both non-coding and not conserved. And in that bubbling pool of fast-mutating junk, they found something incredible. Entirely new regulatory sequences that are unique to us. They'd been forged from sequences that used to be junk DNA. They weren't conserved in other species, so we'd missed them in our previous searches. The researchers call them Human Ancestor Quickly Evolved Regions, or HAQERs. Good work, marketing team. HAQERs were about as unique to humans as it's possible to get. In other species, many of those sequences don't seem to do anything. But in our ancestors' genomes, they'd been converted from junk to functional. And what's more, these HAQERs were the fastest evolved regions of the entire human genome.